Every single living thing on this planet, from a tiny bacterium to a giant blue whale, is built from cells. But what actually makes a cell a cell? Well, it all kicks off with this incredible microscopic barrier that literally separates life from non-life. So today, we're diving deep into that essential structure, the cell membrane.
Okay, so at the heart of it all is the plasma membrane. It's pretty much a universal feature of every cell out there. Think of it as the ultimate gatekeeper, the container for all the chemistry of life, and the communications hub, all rolled into one amazing package. It's what allows a cell to have a controlled internal environment that's totally distinct from the world outside. And what's truly mind-blowing is just how delicate this barrier is. We're talking about a film that's only about 5 nanometers thick. To put that in perspective, that's only about 50 atoms across. But don't let that thinness fool you. This is no static, boring wall. It's a super dynamic, active, and surprisingly fluid interface that is always, always in motion.
So how in the world does a layer that's so thin and fluid manage to hold itself together and act as a stable barrier? Well, the secret is all thanks to the pretty amazing properties of one special type of molecule that builds its basic fabric.
The key word you gotta know here is amphipathic. Yeah, it sounds a little complex, but really, all it means is that the molecule has a kind of split personality when it comes to water. One part absolutely loves water, and the other part, well, it hates it. And this dual nature, this love-hate relationship, is the fundamental reason membranes can just assemble themselves all on their own. Let's check out the classic example, a phospholipid. You see, it's got this polar, hydrophilic head, which is totally attracted to water molecules. And then, attached to that, it has 2 non-polar, hydrophobic tails, which are made of fatty acids. And these tails are repelled by water, kind of like how oil and water just don't mix.
And here's where the magic happens. When you drop these molecules into water, they basically don't have a choice. To get to the most stable, lowest energy state possible, they automatically arrange themselves into this double layer, a bilayer. The water-loving heads face outward, toward the water that's both inside and outside the cell. Meanwhile, the water-hating tails get tucked away into the middle, totally shielded from the water. And just like that, this arrangement forms a self-sealing, closed compartment, the very basis of a cell. Pretty cool, right? But a cell has to do way more than just sit there in a lipid bubble. It needs to interact with the world around it, move materials back and forth, and pick up on signals. So, next up, let's look at the components that give the membrane all of its special functions, the proteins. And this brings us to the big idea for how membranes are structured, the fluid mosaic model. It was proposed back in 1972, and it asks us to picture the membrane not as a rigid structure, but as a 2-dimensional fluid, basically a sea of lipids with all these different protein icebergs just floating around in it. So the main takeaway here is that the proteins aren't just bolted down in one spot. Nope, they can actually drift around laterally within that lipid layer. Some of them, which we call integral proteins, go all the way through the membrane. Others, the peripheral proteins, are just kind of hanging out on one side or the other. This movement, this fluidity, is absolutely crucial for the membrane to do all of its jobs. Okay, so that's a great model, but you know how science works. It's all about the proof. How did scientists actually figure out that these protein icebergs were really moving and not just frozen in place? The answer comes from a really, really clever experiment. So here's what they did. Researchers took a human cell and a mouse cell, and they labeled the unique proteins on each with different fluorescent colors. Let's say green for the human proteins and red for the mouse ones. Then they forced these 2 cells to fuse into one giant hybrid cell. Right after they fused, you could see the red and green proteins were totally separated, each on their own half of the new cell. But then they waited, and after just 40 minutes, they were completely mixed together. And that was it. Direct proof that the proteins had drifted freely through that lipid sea. Now, what's really neat is that a cell can actually control how fluid its membrane is. And it's all about the kinds of lipids it's made of. For example, fatty acid tails that are unsaturated have these little kinks in them, which keeps them from packing together too tightly. And that makes the membrane more fluid. Shorter tails do the same thing. And then there's cholesterol. It's a rigid molecule that wedges itself between the phospholipids. And that makes the membrane a bit stiffer and less fluid. You can think of it like the cell's own biological thermostat. All right, so we've got this fluid, self-sealing lipid bilayer that's studded with all these mobile proteins.
But what are those proteins actually doing for the cell day in and day out? Well, this is where the membrane really comes to life. These proteins are the true workhorses of the membrane. They are not just sitting there as decorations. Each and every one has a very specific job that's absolutely essential for the cell to survive, to grow, and to respond to everything that's happening around it. Let's break it down real quick. You've got transporters, which are basically selective doors and pumps controlling what comes in and what goes out. Then you have anchors, which act like structural rivets, connecting that fragile membrane to a supportive framework inside the cell. Receptors are like the cell's senses. They detect signals from the outside world and then relay that message to the inside. And finally, you've got enzymes, which use the membrane as a handy platform to carry out vital chemical reactions. So, if there's one thing to take away from all this, it's that you have to stop thinking of the cell membrane as just a simple bag or some kind of static wall. That picture is just way too simple for what's really happening. Instead, what we have is a fluid and incredibly dynamic structure. It's asymmetrical, meaning the inside face is different from the outside face. And it's a functional mosaic of all these different lipids and proteins working together in perfect harmony. This elegant, self-assembling boundary is truly what makes a cell, and by extension, all of life, possible.
But all of this leaves us with one last really interesting question. I mean, if the membrane is essentially a two-dimensional liquid, what gives a cell its shape? What's providing the structural support to keep it from just collapsing into a puddle? Well, that is